Welcome to this scientific lecture on liquid-liquid phase separation and membraneless organelles. Today we're going to explore one of the most exciting phenomena in modern cell biology: how cells create functional compartments without membranes. First, let's address a fundamental question. What exactly are membraneless organelles? Traditional organelles, like mitochondria and the nucleus, are surrounded by lipid bilayer membranes that separate their contents from the cytoplasm. However, membraneless organelles form through a completely different mechanism called liquid-liquid phase separation. They exist as liquid droplets within the cellular environment. Examples include nucleoli, which are involved in ribosome biogenesis, stress granules that form under cellular stress, and P-bodies that regulate messenger RNA. Here's a simplified view of phase-separated droplets within a cell. These droplets are dynamic, liquid-like structures that can merge, divide, and exchange components with their surroundings. Now let's dive into the physical chemistry. Why do these droplets form? The answer lies in thermodynamics and polymer physics. Phase separation occurs when it becomes thermodynamically unfavorable for two components to remain mixed. This is similar to oil and water separating. But here's an important question. How do we mathematically describe when phase separation will occur? The answer is Flory-Huggins theory, which was originally developed to describe polymer solutions but applies beautifully to biological phase separation. Let's explore Flory-Huggins theory in detail. This theory gives us a framework to predict phase behavior. The free energy of mixing equals the enthalpy of mixing minus temperature times the entropy of mixing. For polymer solutions, we need to account for the fact that monomers are connected into chains, which affects the entropy. The Flory-Huggins free energy has three terms. The first two terms represent the entropy of mixing. Phi1 over N1 times natural log of Phi1 plus Phi2 over N2 times natural log of Phi2. The third term, Chi times Phi1 times Phi2, represents the enthalpy of mixing. Here, Phi represents the volume fraction of each component in the mixture. N is the degree of polymerization, essentially how many monomers are in each polymer chain. Chi is the Flory-Huggins interaction parameter, which quantifies how favorable or unfavorable the interactions are between different components. Here's the critical question. When does phase separation actually occur? The answer is when Chi exceeds a critical value that depends on the chain length. For unfavorable interactions, Chi is positive and large. The critical Chi equals 1/2 times the quantity 1 over square root of N, 1 plus 1 over square root of N2, all squared. This diagram shows the free energy landscape. When Chi is small, we have a single minimum indicating one homogeneous phase. When Chi is large, we get two minima separated by a barrier, indicating phase separation is favorable. Now let's discuss concentration fluctuations and how droplets actually nucleate and grow. At the molecular level, thermal fluctuations constantly cause local concentration variations throughout the system. But here's a question. How do these tiny fluctuations lead to macroscopic droplets? The answer depends on how deep we are in the phase diagram. There are two mechanisms: spinodal decomposition and nucleation and growth. In spinodal decomposition, which occurs deep in the two-phase region, any fluctuation grows spontaneously without an energy barrier. In nucleation, which occurs near the phase boundary, fluctuations must overcome an energy barrier to form stable droplets. This plot shows how droplet radius grows with time. Initially, there is a lag phase during nucleation, then rapid growth as droplets coarsen through Oswald ripening. Now let's connect this theory to actual biological molecules. What kinds of molecules undergo phase separation in cells? Intrinsically disordered proteins, or IDPs, are key players in biological phase separation. These proteins lack stable secondary structure. Phase separation is driven by multivalent interactions, multiple weak interactions between protein domains or between proteins and RNA. Here's an important question. What types of molecular interactions drive biological phase separation? The answer includes electrostatic interactions between charged residues, pi-pi stacking between aromatic amino acids, and hydrophobic effects from nonpolar residues. This represents the molecular view inside a phase-separated droplet. Disordered proteins shown as red lines interact with RNA molecules shown as green dots through multiple weak interactions. Let's now discuss the material properties of these condensates. They behave as complex fluids with interesting rheological properties. The viscosity of biological condensates varies widely, from water-like to gel-like, depending on the molecular composition and interaction strength. Surface tension at the droplet interface determines whether droplets remain spherical and how readily they fuse with each other. Viscosity often follows an Arrhenius relationship. Et equals Et0 times exponential of activation energy over RT, where viscosity increases with lower temperature. Now let's explore how microfluidics enables precise control over phase separation. This is crucial for creating designer condensates. Microfluidic devices allow exquisite control over the composition, concentration, and mixing of components. We can generate populations of monodispersed droplets with precisely defined properties, something impossible with bulk methods. This schematic shows a microfluidic device. Two channels carry protein solution and crowding agent. They merge at a junction where mixing induces phase separation, generating uniform droplets downstream. Microfluidics also enables rapid tuning of environmental parameters like temperature, pH, and ionic strength, allowing us to map phase diagrams efficiently. So what are the key advantages of microfluidics over traditional bulk experiments? The advantages include dramatically increased experimental speed, much higher precision in controlling conditions, and orders of magnitude less sample consumption. Now let's explore designing synthetic condensates with tailored properties for specific applications. We can now engineer proteins with specific interaction motifs, tuning the strength and valency of interactions to control phase behavior. Elastin-like polypeptides are excellent model systems. They show temperature-responsive phase separation that can be precisely tuned through sequence design. One powerful application is creating designer condensates for biocatalysis, where we can concentrate enzymes and substrates to accelerate reaction rates. This illustrates a catalytic condensate. Enzymes, shown as green E, and substrates, shown as yellow S, are concentrated inside the droplet, dramatically increasing the local reaction rate and producing products shown as cyan P. Let's explore applications in synthetic biology and the construction of artificial cells. Phase-separated compartments provide a route to create functional organization in protocells without the need for lipid membranes. We can create artificial organelles that perform specific functions like concentrating genetic circuits or sequestering metabolic pathways. So why is this particularly useful for synthetic cell construction? The answer is that it provides spatial organization and functional compartmentalization without the complexity of synthesizing and maintaining lipid membranes. Let's discuss phase diagrams in detail. These are essential tools for understanding and predicting phase separation behavior. A phase diagram maps out regions where the system exists as one homogeneous phase versus two separated phases. Here's a typical phase diagram. The red region indicates two-phase behavior where droplets form. The green region is single phase. The boundary is called the binodal, and the yellow dot marks the critical point. The critical point is where the distinction between the two phases vanishes. It marks the boundary beyond which phase separation cannot occur. The critical temperature is proportional to chi times n, the interaction parameter, times the chain length. How do we experimentally determine these phase diagrams? We use turbidity measurements to detect droplet formation, direct microscopy to visualize droplets, and light scattering to quantify droplet size distributions. Now let's examine the kinetics. How fast do these processes occur, and what controls the timescales? Phase separation typically proceeds through three stages: initial nucleation of small droplets, growth of individual droplets, and coarsening, where larger droplets grow at the expense of smaller ones. During late-stage coarsening, the average droplet radius grows as time to the 1/3 power, a signature of Ostwald ripening. This plot shows the characteristic cube root growth law during coarsening. The red points show experimental data following the predicted time to the 1/3 power scaling. Molecules continuously exchange between droplets and the surrounding bulk phase. This exchange rate depends on the binding affinity and diffusion coefficients. The characteristic exchange time equals 1/k off, the dissociation rate constant. Let's discuss advanced strategies for controlling condensate properties and behavior. We can use optogenetic tools to trigger phase separation with light, enabling precise spatio-temporal control. Chemical inducers like small molecules can trigger reversible assembly and disassembly of condensates. An exciting question: can we create condensates that respond to specific signals or environmental changes? Yes, by incorporating responsive protein domains that change their interactions in response to pH, temperature, or specific ligands. This shows light-induced phase separation. Before light exposure, proteins are dispersed. Upon illumination, they oligomerize and phase separate into droplets. Let's review the key experimental techniques used to characterize phase-separated systems. Fluorescence microscopy with labeled proteins allows direct visualization of droplet formation, fusion, and internal dynamics. Fluorescence recovery after photobleaching measures how fast molecules move inside droplets, revealing viscosity and binding kinetics. Dynamic light scattering provides information about droplet size distributions and their temporal evolution. Atomic force microscopy can probe the mechanical properties like elasticity and adhesion of individual droplets. A critical question: how do we rigorously determine if a system is truly undergoing liquid phase separation? Key criteria include spherical droplet morphology indicating liquid-like behavior, droplet fusion demonstrating fluidity, and rapid FRAP recovery showing internal dynamics. Now let's connect this to biology and disease. Phase separation plays crucial roles in cellular function and dysfunction. Stress granules are phase-separated assemblies that form when cells experience stress, sequestering mRNAs and translation factors. Aberrant phase transitions, where liquid droplets convert to solid aggregates, are implicated in neurodegenerative diseases like ALS and Alzheimer's. This raises an important question: how do liquid condensates transition to pathological solid aggregates? The answer involves condensate aging, where over time molecules within droplets adopt more stable, less mobile configurations, eventually forming irreversible fibrils. This illustrates the pathological transition. A healthy liquid droplet on the left can transition over time to a solid fibrillar aggregate on the right, losing its beneficial dynamic properties. Understanding phase separation opens new therapeutic strategies for treating diseases associated with protein aggregation. Small molecules can be designed to modulate the phase separation propensity of disease-associated proteins, either promoting or suppressing droplet formation. Strategies to prevent liquid-to-solid transitions include molecules that maintain droplet fluidity or promote droplet dissolution. Let's look toward the future. What exciting developments lie ahead in designer condensate research? Researchers are creating multi-component condensates with layered internal organization, mimicking the complexity of natural organelles. Catalytic condensates that concentrate entire metabolic pathways could revolutionize metabolic engineering and synthetic biology. Phase-separated droplets could serve as smart drug delivery vehicles that release cargo in response to specific cellular signals. The grand challenge: can we create truly biomimetic artificial cells using phase separation principles? We're getting closer. By combining designer condensates with other synthetic biology tools, we are approaching the goal of building functional artificial cells from the bottom up. Computational approaches are increasingly important for predicting and understanding phase separation. Molecular dynamics simulations can capture atomic-level details of how proteins interact and form condensates, though they are limited to short timescales. Coarse-grained models that represent proteins as simplified entities enable simulations of larger systems and longer timescales. The stickers and spacers model treats proteins as chains with sticky interaction sites separated by flexible spacers, capturing essential physics while remaining computationally tractable. Machine learning approaches are now being trained to predict phase diagrams directly from protein sequences, potentially accelerating condensate design. Let me summarize the key concepts we've covered in this comprehensive lecture. Liquid-liquid phase separation is a fundamental mechanism by which cells create functional compartments without membranes. Flory-Huggins theory provides a quantitative framework for understanding when and why phase separation occurs, based on entropic and enthalpic contributions. In biology, phase separation is driven by multivalent weak interactions between disordered protein regions and nucleic acids. Microfluidic technologies enable unprecedented control over condensate formation and characterization of their properties. Designer condensates have exciting applications spanning chemistry, biology, medicine, and materials science. Aberrant phase transitions from liquid to solid states are implicated in neurodegenerative diseases, offering new therapeutic targets. The field is rapidly advancing toward creating biomimetic artificial cells and organelles using phase separation principles. In conclusion, liquid phase separation represents a paradigm shift in our understanding of cellular organization. We have moved from viewing the cell as a collection of membrane-bound organelles to recognizing the importance of dynamic, liquid-like condensates. The ability to design and control these condensates opens extraordinary possibilities for biotechnology and medicine. Thank you for your attention to this fascinating topic.